Our next question in the physical biology exam is what length of book would a DNA polymerase copy without an error? And I'm thinking about including proofreading. Okay, would it be something like the Bible, the Encyclopedia Britannica, or the English Wikipedia? Okay, and so what's the error rate of the different molecular machines of the central dogma? You can see that for DNA replication, we got on the order of an error rate of 10 to the minus 10 errors per base pair, which means, that for DNA polymerase to make an error, on average, it needs to copy 10 to the 10 base pairs. Okay, so let's figure out how many characters there are in each one of these three options and see to get a sense of how long of a book that would correspond to without making any mistakes whatsoever. Okay, and the first thing I want to do is to estimate. So estimate the number of letters per page. The typical number of letters per page. Okay, and I don't know, what would you say? You would say something like, in a typical page, I would say that there's a few tens of lines per page. And there's on the order of 10 words per page, right? And let's just say that there's on the order of a few letters per word, right? So this is what we will call the number of in a page, the number of letters. Okay, so few times few, that's 10. Then I have 10 lines. Then I have 10 words per page. So this is letters per page, which is on the order of 10 to the 3 letters per page. Okay, so now let's figure it out, for example, for the Bible. Okay, and so number of letters in the Bible. So how many pages does it have? Let's say that it has on the order of a few thousand pages. And then we have 10 to the 3 letters per page, which gives us a few million letters. And it's a pretty good estimate, as you can see here. There's on the order of 10 to the 7 letters in the Bible. At least I think that this is the King James edition. Okay, so we're within a factor of a few in our estimation, and clearly DNA polymerase could make a copy of this without making any mistakes whatsoever. All right, so now let's figure out the here this was number of letters in the Bible. So now let's figure out the number of letters in the encyclopedia. So in this case, I don't know, and encyclopedia letters. I guess I would say that there's, how many tomes are there? Like there's on the order of, maybe a few times 10 tomes. Tomes. Each one containing on the order of 1,000 pages per tome. And as our estimate also tells us that we got 10 to the 3 letters per page. So this tells us that there's on the order of a few times 10 to the 6, 10 to the 7. letters. And once again, let me see if I can get this right. If we look at this, we're within an order of magnitude. Like, if you do you can see here what I said is, yeah, that there's, here I estimate, it's the actual is on the order of a few times 10 to the 8. So here we're not asked, we didn't do as well. But then now let's do an estimate of, it was right. Yeah, I think I did this right. So now how do I estimate the number of letters in Wikipedia? And this was a little bit harder to do. So what I decided to do is to say that the number of letters in Wikipedia, the letters, is 100-fold more than the number of letters in the encyclopedia. And so this gives us a few, from our estimate, times 10 to the 9 letters, which, if you look at it, we're within an order of magnitude. Like if you look at actual Wikipedia, you get a few times 10 to the 10, as shown here. So what does this tell us? That, DNA polymerase would basically copy all of Wikipedia and make one error, right? And this is crazy. It's the fidelity of this machine is crazy. And Tanya Baker put it in this great article, put it in such a nice way, which I encourage you to try to maybe try to redo her calculation. But the idea that they try to do is to scale up these rates to get a sense of how wonderful of a machine DNA polymerase is. And, so what she said is synthesis of all genomic DNA involves the highly coordinated action of multiple polypeptides. These proteins assemble 2 new DNA chains at a remarkable pace, approaching 1,000 nucleotides per second in E. coli. Okay, so if the DNA duplex were 1 meter in diameter, then the following statement would roughly describe E. coli replication. The fork would move at approximately 600 kilometers per hour, or 375 miles per hour, and the replication machinery would be about the size of a FedEx delivery truck. Replicating the E. coli genome would be a 40-minute, 400-kilometer trip for 2 such machines, which would on average make an error only once every 170 kilometers. I mean, it's just amazing what these machines can do. And this has to do also with a whole set of interesting questions related to the proofreading, to how you ensure fidelity. So, for example, one of the great stories that we will explore is that of the fidelity in translation. Because when you do a simple model based on statistical mechanics, asking, how often should translation screw up? Okay, because you get either 3 or 2 hydrogen bonds in the incorporation of a new amino acid, you expect that the error rate should be 1 in 10 or 1 in 100. But actually, if we go up here, you see that the error in translation is a lot higher. It's a lot lower, sorry. Right? We're talking about 10 to the 5 or so. Okay? And how does this happen? Well, it turns out that you can use energy to ensure fidelity. You can use energy to field error correction. And this is this great idea of so-called kinetic proofreading that was developed by John Hopfield and Jack Nino. And, when we talk about defying equilibrium, spending energy to defy equilibrium, we'll go back to this fascinating problem.